Hello, my name is Toshiya Tanaka. Research collaborators are Shigito Seno and Hideo Matsuda. We are at Osaka University and, today we will talk about feature selection with VAE for scRNA-seq analysis. Background and purpose. Single-cell analysis technology has made rapid progress in recent years. It has provided many valuable insights into biological systems, such as early disease diagnosis in cancer. Clustering cells is significant to grasp the relationships between each cell. For example, it allows building the trajectory inference. When clustering cells with gene expression levels data, choosing irrelevant features is essential. Principal component analysis. PCA is one of the most common methods for feature selection. PCA is strong but is affected by deviations in the amount of each cell type. TSNE is suitable for visualization. However, each of the reduced dimensions by TSNE has little meaning to the original data. We took on the challenge of learning the representations of the features by variational autoencoder, VAE. Each dimension in a latent space of VAE retains meaningful properties of the original data because VAE reconstructs the original data from the low-dimensional representation. There are a lot of bioinformatics methods for analyzing and interpreting gene expression data. Most of them require several individual data processing steps. The differences could change the results. To avoid it, using raw data as inputs in our method. Method. Let x be the input data and z be the latent variables of x. VAEs encode data x into z and decode the latent variable z to the data x. VAE learns the good representation z by repeating this process over and over again. The goal of VAE is to maximize the likelihood px. However, since it is almost impossible to calculate px directly, we use evidence lower bound, ELBO, to avoid the problem. ELBO is the approximation of the likelihood px. ELBO represents as follows. This ELBO is different from normal VAEs. It is for the Gaussian mixture model VAE. The probability distribution of the latent variable of normal VAEs is a Gaussian distribution. However, it cannot represent multiple modes of different cell types in scRNA-seq data. Therefore, we use the Gaussian mixture model instead of the Gaussian distribution. The VAE is a categorical variable that shows the mixing probability of each cluster. The larger the negative reconstruction loss and the smaller Kullback-Leibler divergence, the more maximized ELBO is given. After training the VAE, latent variable z grasps the important features of input data x. Results. We tested k-means clustering cells with latent representations and measuring the adjusted rand index, ARI, as clustering metrics. We used 2 data sets and reduced the number of dimensions of each data to 5. Below are 2 results for each data set and ARI comparisons between 3 clustering methods. The images on the left are the latent space visualized by TSNE. The images on the right show the values, feature selection, of each dimension in the latent space. Let me explain the results of Data 1 first. Data 1 is single-cell RNA-seq from human lung adenocarcinoma cell lines. 3,918 cells, 11,786 genes, 5 classes. Look at the image on the right. This is the dimensionality reduction result of gene expression data from 11,786 dimensions to 5 dimensions using VAE latent variables. Each cell type shows its value in each dimension and it is neatly divided. The image on the left is the latent space, 5 dimensions, visualized by TSNE. reduced the number of dimensions from 5 to 2. Each cell is colored by its cell type label. We use k-means to cluster the latent space and the reduced data by PCA. Both number of diamond ions is 5. Then, to compare with existing methods, we performed clustering by scRNA-seq. The scores are like this. Data 2 is not single cells but bulk RNA-seq from pure populations of human immune cells, 1561 cells, 48,044 genes, 15 classes. The result looks good, but ARI is not very good. Conclusion. We grasped the meaningful representations of the original data. However, it seems possible that each dimension of the latent space retains more meaningful properties of each cell type. By fixing one feature in the input data and randomly varying the other features, it would be possible to measure the importance of the feature quantitatively, as in factor VAE. This VAE can also be applied to anomaly detection, such as doublet detection. This is all for my presentation. I will further pursue this topic. Sorry for my poor English and thank you for watching.